Good evening. I'd like to welcome everyone to the first annual UPenn Neuroscience Public Lecture. Thank you all for coming. This event is part of Brain Awareness Week, which is an annual week-long event that brings together scientists, clinicians, educators, and journalists to raise awareness about the progress and benefits of brain research. In addition to tonight's lecture, there's some very cool events going on at the Franklin Institute, a lot of hands-on activities, stuff for kids. Dr. Jayatri Das, she's the chief bioscientist at the Franklin Institute. Is she here? There you are. She can stand up for a second. She'll be here. She'll be here with us tonight after the program, and she'll be happy to tell you either about the events tomorrow or the upcoming Your Brain exhibit, which opens this June at the Franklin Institute. Okay. Before we get started, though, I'd like to give you all a brief, very brief history a brief overview of the history of neuroscience. Okay. 25,000 years ago, in the late Stone Age, early humans performed trepanation, which is the practice of drilling holes into the skull, and it was a surgical procedure used to cure mental ailments. The bone that was trepaned out of the skull was kept and worn by prehistoric people as a charm to ward off evil spirits. And trepanation is actually the oldest documented surgical procedure for which we have archaeological evidence. Also of note, this surgical procedure was done up until the 1600s in Europe, just to give you an idea of how far we've come. So, thousands of years later, the ancient Egyptians were really the first to rigorously study the brain. They routinely extracted the brain in preparation for mummification using surgical tools that are seen, like those seen here. And what we have here, the Edwin Smith Papyrus, is actually a collection of 48 case studies that contains realistic anatomical, physiological, and pathological observations of the major organs of the body. It actually contains the first known description of the anatomy of the human brain. And as a matter of fact, the word brain was used for the very first time in written language in this papyrus. However, despite these meticulous observations, the ancient Egyptians believed that the heart, not the brain, was the source of consciousness and intelligence. The Greek physician Hippocrates, he was the first to challenge this view, and he believed that the brain was not only responsible for sensory perception, but also was the seat of human intelligence. And he described his observations in his biggest work, I would say, the Corpus Hippocratum. He's also the guy that the Hippocratic Oath was named after. However, he didn't have the necessary scientific tools or techniques to really bolster the evidence to his claim. So, consequently, for nearly 2,000 years, the study of the brain was relegated primarily to the area of philosophy. Then, in the late 19th century, modern neuroscience was born with the pioneering works of neuroscientists Santiago Ramon y Cajal and Camilo Golgi. At the time that these two scientists were active, this is the late 1800s, there was an ongoing debate about the organization and architecture of the brain. One camp of researchers, which included Golgi, believed that the brain consisted of a large network of tissue, which they called the reticulum, and that there were no really discrete elements of the brain. The other camp, which Cajal was a part of, believed that the brain was made up of distinct elements or cells, that he believed. Cajal, using a staining technique that Golgi actually developed, he was able to identify individual brain cells, which he called neurons. This monumental achievement led to the realization that the neuron, a cell, is the functional unit of the human brain, which ended the debate between Cajal and Golgi. What's ironic, though, is that Cajal could have never done this work except for Golgi's staining technique, which is still used today. So, in 1906, Cajal and Golgi both shared the Nobel Prize for their work. And these are some of Cajal's drawings of neurons that he observed after staining under a microscope. In the second half of the 20th century, major advances in molecular biology, like the discovery of the DNA, as well as major advances in electrophysiology, which is recording the electrical activity of neurons, they sparked a vigorous study of the brain. Currently, neuroscience is a highly interdisciplinary science encompassing the fields of biology, medicine, chemistry, physics, computer science, mathematics, linguistics, psychology, philosophy, you name it. Over the past several decades, neuroscientists have discovered a vast amount about the human brain. Among many of the groundbreaking findings, neuroscientists have discovered where and how memories are stored in the brain. We've created brain imaging tools to see the brain's response to sight, sounds, and other sensations. And they've elucidated some of the biological basis of many of the psychiatric disorders and have developed or begun to develop pharmacological treatments for some of these disorders. However, despite all the advances that we have with neuroscience research, there's still a whole lot that we don't know. Here at Penn, we have some fantastic research going on, and an event like Brain Awareness Week gives us the opportunity to share some of that research with you guys. We got a great program for you all tonight. Tonight we'll hear from 4 world-renowned faculty members, and they didn't pay me 20 bucks to say that. That's actually true. We'll hear from 4 world-renowned faculty members about the latest advances in research on obesity, decision-making, drug addiction, and neuroengineering. After the program, there's a reception, and you're all, of course, invited to stay, and it'll be held just outside the auditorium here. So, without any further hesitation, I'd like to introduce our first speaker, Teresa Reyes. She's a research assistant professor here, and she's affiliated with both the neuroscience and pharmacology graduate groups. She got her BS, MS, and PhD all in the Department of Psychology at the University of Wisconsin in Madison. Her PhD in biological psychiatry focused on neuroimmunology, and during her postdoctoral training at the Salk Institute in La Jolla, California, she studied functional neuroanatomy. Her research interests focus on how prenatal and postnatal diet can impact development and brain functions, especially in the pathways that regulate appetite and reward. Her work has been featured in many top-tier journals, as well as in the popular media, including National Public Radio. Let's give a warm round of applause for Dr. Reyes.
Super. Thank you very much all for coming. I'm actually really thrilled to be participating in a Brain Awareness Week event. I've done 2 other events this week in my kids' schools. So earlier this week, I talked to a pre-K class about what the brain does in the morning. In the afternoon, I gave a couple of lectures to a series of 3rd grade classes. So we're moving up. We did pre-K, 3rd grade. So hopefully I've moved past that and I'm directing this at the right audience. Okay, so I'm just going to tell you 2 sort of short stories about some of the work that goes on in my lab. And this is really what I care about, which is how what we eat affects how our brain functions, brain development, and eventually behavior. Okay, so this is sort of the grounding of what I'm interested in. And this is a map showing the prevalence of obesity. And what you can see is that this is a statistic we're all familiar with, the prevalence rates of obesity starting in 1999, advancing through time to 2009, has rapidly increased. So there's a much greater percentage of the population that is overweight or obese. We're particularly interested in how this obesity epidemic affects particular time periods that are critical periods of brain development. So the first period that I'm going to talk about is how maternal obesity during pregnancy can affect offspring brain development. And the second period of time that is critical, sort of a critical brain development time period, is adolescence. So how does obesity during adolescence affect brain function and behavior? So let's first talk about the epidemic of maternal obesity. So in 2009, these are the numbers of women of childbearing age that were overweight or obese, so that you can see 29% of this population was overweight and 24% of this population was obese. So the majority of women of childbearing age were either overweight or obese. And when you actually look at the number of women who are obese or overweight who really were pregnant, that characterizes half of the women. So this is really something that affects the majority of babies who are born in this country. Along with obesity during pregnancy is a related condition, which is excessive gestational weight gain. So physicians have guidelines for how much weight is healthy for a woman to gain during pregnancy, and that's based on what their body weight was prior to pregnancy. We know that half of women who were normal weight prior to pregnancy gained too much weight during pregnancy, and 3/4 of women who had a high BMI prior to pregnancy gained too much weight during pregnancy. So these pregnancy conditions, obesity during pregnancy or gaining too much weight during pregnancy, again affect the majority of babies that are born in this country. So why is that a concern? Well, partly it's because babies who are born to moms who are obese or whose moms gain too much weight during pregnancy are at a much greater risk of being born large for gestational age, which is this LGA. So they're 3 times as likely to be LGA. And LGA babies are at an increased risk for obesity themselves. Those babies are more likely to develop more severe obesity, as well as learning and or behavioral problems later on. And so we were really interested in trying to model this in mice so that we could understand exactly how it is that the brain is changed in a way that might predispose these babies to the development of either obesity or behavioral or learning deficits later in life. So we use a mouse model where mice are fed a high-fat diet during pregnancy and lactation. So these mice are actually normal weight at the time of breeding, and they gain too much weight during pregnancy. So we're able to model this in mice. And just like in humans, these pups are born large for gestational age, and they maintain that heavier body weight throughout the period of lactation. And once they're weaned, their body weight eventually normalizes. Okay, so there are a lot of systems in the brain that control how we eat food. One of these is the sort of homeostatic system that regulates our food intake that is based on our metabolic needs, so eating when we are hungry. But there is this other system within the brain that sort of drives us to eat for reasons that are not related to our metabolic needs. So this is eating when it exceeds your metabolic needs. So eating because it's tasty, or because it's sweet or salty or sugary or fatty, or you're bored, or you're out for dinner, or you're tired, or all these reasons that we tend to eat that are not related to just being hungry. So the question was, and it's really this hedonic eating, which tends to be very calorie-dense types of food, that is really going to predispose for obesity or set up this obesity risk. So what we did was to look at the offspring once they were adults. So the moms ate a high-fat diet during pregnancy and lactation, and then we looked at the adult offspring. It's a very simple task in which we give the mice a choice to drink sucrose or water, or to eat a high-fat diet or a chow diet. All animals prefer either the sweetened solution or the high-fat diet, so that's a preference over 50%. But if the mom ate a high-fat diet during pregnancy and lactation, those offspring as adults have a significantly stronger preference for that sugary solution and that high-fat food. Okay, so then we wanted to look at some of the circuits within the brain to see if we could better understand how the brain is changed in a way that would underlie these behaviors. So we have this circuitry in the brain that tells us when something is rewarding, things like food, social interaction, for some of us exercise, for some of us when we're done exercising. And then this sort of system is what becomes dysregulated or co-opted by drugs of abuse. And there's just one example that I'm going to show you today, which is the opioids. So opioids are molecules that function within this reward circuitry. And when we looked at the expression of opioid molecules, so this is one of the ligands and this is a receptor, what you can see is that there was this significant dysregulation of this system in the offspring brain if the mom ate a high-fat diet during pregnancy and lactation. Okay, so let me just show you a short story about some of the other sort of neurobehavioral and cognitive learning endpoints that we've looked at. The reason we've done that is that there is data in the literature to support the idea that maternal obesity or pre-pregnancy BMI can also set up a risk in the offspring for cognitive or behavioral deficits. So to do this in the mice, we use a 9-hole mouse operant chamber. The mice, and it's amazing, they really do learn this task. They have sort of 5 holes that are activated, or that are present, and there's a light that will light up. And what the animal eventually learns is that when the light comes on in a specific hole, they need to go and nose poke in that hole, and then they will get rewarded with a little drop of chocolate yoo-hoo. And so I'm going to show you data from 2 tasks. One is called the progressive ratio, which is a measure of motivation, in which we can see how hard an animal is willing to work for that sweetened reward. And the second task is a test of executive function, which will let us look at things like attention and impulsivity. Okay, so here's what the animal has to do now in this task, is that they have to nose poke an increasing number of times to get the same single reward. So the first time they nose poke once, and they get their single drop of yoo-hoo, but the next time they have to nose poke 2 times, then 4 times, then 9 times. And so how hard an animal is willing to work is a measure of their motivation, because at some point an animal will say, I am not nose poking 27 times for my 1 drop of yoo-hoo. So what you can see here is that if the moms ate a high-fat diet during pregnancy and lactation, these offspring are significantly less motivated to work for the food, for the reward. And so remember, it's these animals that actually have a significantly stronger preference. So when they don't have to work for the sweetened food, they prefer it. So this is not just a function of them not finding it rewarding, but if they have to sort of act in a motivated way to acquire that behavior, to acquire that reward, they are less willing to do it. So they're kind of lazy. Let me just show you some data from the executive function tasks. So here, there are now 5 different holes. The animals have a choice across these 5 holes. And what they have to do is attend to the light coming on. And when the light comes on, they nose poke in that and they get their reward. So there are a lot of ways they can make an incorrect response. They can omit, the light comes on, they don't do anything. They can make an incorrect response to the hole that is not lit, or before the light even comes on, they can make a response. Omissions are a sign of inattention, while incorrect and premature responses are indications of impulsive-like behavior. So what we see in these LGA offspring is that they make significantly fewer correct responses, so they are not as good at doing the task. And when we look at what kind of mistakes they make, we see that there's no difference in their omitted responses. So it's not that they are inattentive. But if you look at their incorrects and prematures, these animals are making errors that look like impulsivity. So if the mom ate a high-fat diet during pregnancy and lactation, we see behaviors that look like impulsivity in those offspring when they're adults. We can also look at how quickly they learned the task. And what you can see, these are the LGAs here in this blue line, is that after about 12 days, all of the control animals had learned the task. But it took nearly 3 times as long. It took 30 days for all of the animals from the high-fat fed moms to learn the task. So in general, they were significantly slower to learn it. Okay, and lastly, let me just talk a little bit about adolescence. So now we are out of the prenatal vulnerability, and we're talking about consumption of a high-fat diet postnatally. The reason that this is important and that we care about it is that there's an increasing amount of data that shows that the chronic intake of highly palatable foods, high-fat diet, high-sugar diet, repeatedly is activating this reward system, and that repeated activation can change the way the brain responds to these rewarding stimuli. We've seen it in animals, and we've also seen it in humans. So if you take This was done in college students, and you put them in, for example, a functional magnetic resonance imaging scanner, and look at how those people who are either obese or normal weight respond to a palatable milkshake. Subjects who are overweight or obese have a significantly blunted response to that milkshake. Okay, so the question that we were interested in asking was, we know that there are changes in this reward circuitry in response to a chronic high-fat diet. But is there sort of a critical period? So if that exposure happens during adolescence, is the adolescent brain more vulnerable? And are these changes reversible if you just take away the diet? This is the work of one of my graduate students who's defending her thesis in days. So she had these were her ideas, which were just great ideas. And this is essentially the experimental design, which is this, that animals were on a high-fat diet for 12 weeks, but it either started when they were 3 weeks of age, which would cover the period of adolescence, or it started when they were 6 weeks of age, which is more the point of young adulthood. They were all groups were on the diet for 12 weeks, and then we took them off the diet. Another group were taken off the diet for 4 weeks, and then there were animals who were age matched so that we had them at the correct ages. Okay, and I'm now going to introduce this concept of sex differences, which is that men and women are different. Okay, so here's what happens in a male brain. When these animals eat a high-fat diet over this 12-week period of time, and I'm just going to show you sort of snapshots. So this is gene expression within one area of the brain for all animals that started the diet at 3 weeks of age. So here you see this downregulation of these molecules related to reward in this area of the brain in male animals that have consumed the diet for 12 weeks. And interestingly, you see the exact same response in females. So in this way, males and females are not different. So then the question was, if we take the animals off the high-fat diet, can they recover? And actually, I was really surprised because they do recover, even though in 4 weeks, their body weight has not recovered to normal control levels. So these animals are still obese. We've removed the high-fat diet, but now these changes in their brain circuitry have normalized. So that is really great news if you are a male. Because that does not happen in the female brain. I don't know that it doesn't ever happen. It might be that the female brain is slower to recover and that maybe by 6 weeks or 8 weeks, we would see that recovery. But in this brain region at this time period, we didn't see recovery. So now when we look at the difference between 3 weeks and 6 weeks of age, again, the sort of genes that we looked at isn't particularly important, but you can see that there's this downregulation in response to the high-fat diet at 3 weeks of age. And when the diet was started as adults, you see this recovery when you removed the diet. But in this brain region and in these genes, if the diet started as an adolescent, and now these are males, the diet started when the animals were adolescents, this brain region was not able to recover. So in this way, the adolescent brain does appear to be more vulnerable to exposure to the high-fat diet. Okay, this is my last slide. This is what I care about, nutrition and brain development. And so I hope that what I was able to convey to you is that what the mom eats during pregnancy is really important, not only for her own health, but for the health of her baby, and particularly for the brain development of that baby. And what we eat throughout adolescence and even as adults can affect the way our brain works and functions and behaves. And so it's important. These are the people who did the work and the people who pay me money to do the work. So thank you very much.
We have about 5 minutes for questions, and we have microphones on either side if you guys want to chime in.
So you were very meticulous in your talk about mentioning that the moms were fed a high-fat diet during gestation and lactation period. Has there been any further study to see, like, if only gestation or only lactation period can affect the brain development as well?
Yeah, it's a great question. And the reason that we do gestation through lactation in rodents is that we want to try to more fully match what happens in a human pregnancy. and brain development in the rodent. So rodents are born with a more immature brain, so that the first 3 weeks of life is more akin to the third trimester of a human. So that was why we did that. But in these rodent studies, there have been other groups who've split out the lactational period versus just the gestational period, and it's very clear that what happens in lactation also is important in its own right. We've also done work to look at whether or not obesity prior to pregnancy, in the absence of obesity during pregnancy, can have any of these adverse effects on the brain, and it does. So that risk can be imparted even prior to pregnancy.
You mentioned interesting sex differences in your adolescent studies, and I was wondering if you found any of those sex differences in your prenatal studies.
We have. So really, just about anything you do in prenatal work, you will get an interesting sex difference. The behavioral work, though, the attentional, the impulsivity that we've seen, we see that in our female offspring as well. And we see, so actually in those endpoints, they actually are very similar, as well as the preference data. Although in the females, we see a much stronger preference to sucrose and not so much the fat preference, whereas we see both fat and sucrose in the males. So there are some differences. And we see more sex differences in some of the endpoints that we look at that relate to obesity, so metabolic rate and sort of basal food intake and activity, those sorts of things.
Thank you.
Hi, thanks. I was wondering if you've taken this out to further generations, the prenatal stuff. Have you looked into the grandchildren of these mice and seen if the effect lasts if you take the children and keep them on a regular diet?
Yeah, exactly. Super interesting area of research. We have not done it yet. We think about it. We actually thought about one study to do with, we had just a cohort of very obese males that still existed in the lab. So we said, hey, let's just see, if we can look at male, transmission of this phenotype. And I think we bred 8 obese males and 8 normal weight males, and none of the obese males sired pregnancies. So it was like, okay. They were very old though too, so that was so old. But that in itself was sort of like, well, that's interesting. We should probably follow that up. But so it's something that we think about. But there are other groups that are doing that.
I think we have time for one more question.
Thank you for your talk, Teresa. My question was, what are your thoughts on placental reprogramming as a possible link between what mom eats actually is important for the brain development of the pup?
Yeah, the placenta is a really fascinating organ, obviously, that develops only during pregnancy, but it's, the point of communication between what's in the mom and how that gets translated to the baby. And so there is a lot of really interesting work showing how the placenta changes in response to maternal high-fat diet. And there are very clear changes that involve oxidative stress, and there's even morphological changes. And so I think that's an important conduit that could be transmitting this phenotype. But I do think that there's also probably specific components like fatty acids in the diet that are actually getting to the baby, so that it's not just mediated by the placenta, but placental dysfunction is part of the phenotype. Okay, thanks.
Okay. Our next speaker is Dr. Joe Kable, and he is the Baird Term Assistant Professor of Psychology at Penn. He completed his bachelor's degree in chemistry at Emory University and received his PhD right here at Penn in the Neuroscience Graduate Group. His lab studies how people make choices and the neurophysiological mechanisms underlying decision-making. This work combines approaches from experimental economics, the psychology of judgment and decision-making, as well as social and cognitive neuroscience, to help understand how people make trade-offs between the present and the future. He has several publications in highly respected journals, and his work has been featured in the New York Times. Let's give a warm welcome to Dr. Joe Kable.
Hey, thanks for having me here, and thanks for that great introduction. So I want to talk to you today about decision making, and specifically I want to talk to you today about why people quit, why they give up on the long-term goals that they've been working towards, often in favor of short-term rewards that had always been available to them. Now, this is a behavior that will be familiar to any of you who've already made and broken your 2014 New Year's resolutions. It will also be a behavior that's familiar to anyone who, like me, struggles with this dilemma, which is that I'd like to have washboard abs, but I also have a very wicked sweet tooth. Now, from a decision theory perspective, we should recognize at the outset that there would be nothing inherently irrational about prioritizing one of these goals over the other. If I decided I wanted to restrict my caloric intake in order to work towards those washboard abs, that would be fine. If I decided that a couple of extra pounds was fine in order to enjoy the moment and get to have dessert, that would be fine as well. But it is a little puzzling when I say, I'm going to start a diet and I'm going to work towards those washboard abs, and I persist in that choice for months or weeks or days, and then, dessert comes around and I have a look at the chocolate cake and I say, yeah, all that stuff I said about the diet, forget about it. So this is, this is the behavior that we'd like to understand. And it's puzzling because if I was just going to have the chocolate cake in the end, why didn't I save myself all the effort of persisting in this choice if I wasn't going to reach my goal in the end of it? Now, if you look in our culture, the explanation for this kind of behavior is surprisingly one that strangely enough, both St. Augustine and Freud would agree on. And the explanation they would give you is that there's some fundamental limitation in the human cognitive mechanism that keeps us from being able to reach our long-term goals. Whether that's, the flesh being weak, or whether that's the id, or whether that's a self-control strength that depletes with time, they believe in a fundamental limitation. I'm going to hope to convince you of an alternative view that recognizes that there can be hidden rationalities in quitting, that looks at the function that behavior might be trying to serve. And I think by taking that viewpoint, we come to some novel suggestions about how we might be able to encourage persistence in those situations where we'd like to see it. Now, our work takes as a jumping off point a set of experiments by a psychologist, Walter Mischel. Many of you may have heard of these experiments. What he did in this set of experiments was take a 4-year-old, bring them into a room, set in front of that child 2 rewards, one that's more preferred, say 3 marshmallows, and another that's less preferred, say 1 marshmallow, and say, you can have the more preferred reward if, now I'm going to leave the room now, and if you can wait until I come back, you can have the more preferred reward. But sometimes I'm gone for a really long time, and I understand that, and if you're ever sick of waiting, you can call me back and you can have the 1 marshmallow instead. And what he sees, there's lots of very cute YouTube videos if you want to see this experiment online. What he sees is that all the kids start waiting for the delayed reward, and they try their hardest to wait it out until the experimenter comes back, but the majority of them give in and bring the experimenter back in and take the 1 marshmallow instead in the end. Now, we think that the temporal structure of this experiment captures the temporal structure of the kind of persistence dilemmas that many of us face in the real life. Because for the kid in Michelle's experiments, those 3 marshmallows, they don't know when the experimenter's coming back. They don't know when they're going to reach that goal. But that 1 marshmallow is always available immediately to them. And I think this maps on to dilemmas like my dilemma. When I start that diet, I don't know when I'm going to have washboard abs. But in the world that we live in, chocolate cake is pretty much always immediately available to me with a little bit of effort. So what should you do when you face this kind of intertemporal choice dilemma? It turns out that what you should do in this dilemma hinges critically on what you believe are the possible times that that reward that you're waiting for might arrive at. Now, to talk you through it, I'm going to show you a couple of movies. So one possibility is that you might believe that the awaited reward is going to occur at a very specific point in time, and maybe you're a little bit off, so maybe it's likely to come at that time, but it could come a little bit earlier, a little bit later. Another way of saying that is that you have a Gaussian belief about the time at which that delayed reward is going to occur. It's most likely here, but it could happen a little earlier or a little later with less probability. I'm plotting here the probability density as a function of time. Now, what I'm going to show you is how, so already illustrated here is your prediction about when the reward occurs. That's the gold line. And I'm going to show you how that prediction about when the reward occurs changes as you wait through time and rule out all those early possibilities that you thought had some lesser probability. And I'm also going to show you how the distance between you, which is going to be illustrated with a black bar, and your predicted time of reward, which is illustrated with this gold bar, changes as a function of time. The amount of time you predict that you have remaining until you get that awaited goal. And what you see is that as you move through time, if you start with Gaussian beliefs and you rule out those early time periods, well, the reward hasn't come yet, your predicted time of when the reward does occur is going to get pushed farther and farther back. But you're always getting closer to it. The distance between you and that reward, the amount of time you think you have left, is always getting smaller as you wait. And this is our intuition about waiting. If we think something's coming in 3 minutes and we've waited 1 minute, we're closer to it. However, a very different situation obtains if instead you start with a heavy-tailed belief about when that awaited reward will occur. And what I mean by heavy-tailed is, what I mean by heavy-tailed is that there's a large probability, there's some probability the reward will come very soon, and then also a non-zero probability that the reward will also come at some very later point in time. And here what you see is that as you move through time, the reward is getting farther and farther away from you. The predicted time you have left remaining is increasing as a function of the time that you've waited. The intuition here is that in this world, the reward could come quickly, or it could come at a very long time constant. And the longer that you wait without the reward coming, the more likely it is that you're in the long world as opposed to the short world. Now, this is a very nice mathematical idea, and it could explain why people quit when they're waiting for their delayed rewards to pay off. Because if you started out with heavy-tailed beliefs, you'd reach some point where that reward is getting farther and farther and farther away, and now, even though it was worth waiting for initially, it's no longer worth waiting for. And this idea could explain quitting, but do people really hold heavy-tailed expectations? Now, one reason people might hold heavy-tailed expectations is because the distribution of payoff times is a heavy tail. My favorite example of something in the real world that has a heavy-tailed distribution is the time at which someone's going to respond to your email. And I think we all have reached that point in time when we're no longer waiting for the response to come in. The other reason why people might have heavy-tailed expectations is because that's the set of expectations you should have if you're given no other information. And there are theoretical arguments for that. But do people actually hold them? So the first thing we did was ask people, what are your expectations? We said, you've been waiting in a bunch of situations that commonly are thought to involve self-control. You've been waiting for a delayed reward to occur. You've been waiting this long. How much longer do you think it's going to be? So, for example, we asked people about the diet situation. You've been on a diet for a certain period of time. It hasn't worked yet. How much longer do you think it's going to be until it pays off? And what we see is the time that they predict they have remaining until it pays off increases as a function of the time that we say you've been working towards it without success. And this is the signature of starting out with heavy-tailed expectations. If you remember that movie, time left increases as a function of time elapsed. Similarly, if we ask people about practicing the piano in order to play a piece without mistakes, or you're running to improve your running time, or you're studying to improve your score, the amount of time that you think you have left until you reach that goal increases as a function of the time that you've been working towards that goal without seeing results. This suggests that one reason why people may have trouble persisting in these situations is because their expectations tell them that unlimited persistence is not a good thing. The next question we wanted to ask was whether people can learn the statistics of the environment and adjust persistence accordingly. So can we change this? If people start out with heavy tail beliefs or they start out with Gaussian beliefs, can we change it? So having measured temporal beliefs, now we're going to try to manipulate them. And to do that, we brought subjects into the lab and had them do a cognitive task. And this task was very simple. Subjects on each trial of the experiment were given a token. The token was initially worth nothing, but all of the tokens would eventually mature to be worth some money. And if you waited until that token matured, you could sell it back to us and get money. However, if at any point in time you were sick and tired of waiting for that particular token to mature, you could sell it back to us, not get any money, but you could get another token. And your goal in this experiment was to try and make as much money as possible throughout the course of the experiment by selling tokens back to the experimenter. Now, subjects faced two different payoff distributions of tokens. In one situation, the payoff distribution was uniform. So there was an equal probability of the token paying off from 5, 10, 15, 20 seconds, and so forth. In the other condition, the payoff distribution was heavy-tailed. So half of the time, the tokens paid off before 20 seconds, and the other half of the time, they only paid off after waiting for 90 seconds. And the tokens in the two conditions were denoted with different colors, and the subjects played with one color token for 10 minutes, and then played with another color token for another 10 minutes, and so on and so forth throughout the experiment. Now, the strategy that you should use for these two kinds of tokens is dramatically different. For the uniform tokens, your strategy should be to never, ever, ever give up on waiting. Because once you've waited a little bit of time, you should always wait until all of the tokens pay off. Your strategy in the heavy-tailed condition should not be to be persistent for an unlimited period of time, but rather your strategy should be to wait for 20 seconds and then give up if the token hasn't paid off and move on to the next one. So our first question was whether people learned that from experience. From experience with the temporal statistics, did they learn to be as persistent as possible in one environment and to have limited persistence in the other environment? And what I'm showing here is the survival rate, the likelihood that the subject's still waiting for the token to pay off as a function of elapsed time in the two environments. What you see is there's very little difference over the first 15 seconds or so. People are pretty persistent in both the uniform and the heavy-tailed environment. But after that, persistence drops off dramatically. For the purple tokens in the heavy-tailed environment, people are mostly quitting after about 20 seconds, whereas persistence is maintained in the uniform environment. People are mostly staying put and waiting all the way through the end for the green tokens. Now, the subjects who are doing this experiment were actually doing it in an fMRI scanner, so we could also look at their brain activity while they were in the two conditions. And the question that we wanted to ask about the brain activity was whether there's neural evidence that people are dynamically reevaluating rewards as time passes. My suggestion to you is that as people wait, they're continually reassessing how valuable would it be for me to continue to wait for this delayed reward versus how valuable would it be for me to quit and move on to the next trial. And can we get evidence for that? Well, the two things that we would need in order to test that idea, one is we'd need to have a brain region where activity reflected someone's evaluation of their chosen item. And it turns out that there are lots of imaging studies now, and we've done a meta-analysis of over 200 of them, that suggests that this region of the brain, the ventromedial prefrontal cortex, shown here, that the brain activity that we can measure with fMRI, that brain activity reflects how positively someone evaluates the item that they've currently chosen. In addition, to test this idea, we need not just a place to look, but also what to look for. And here we can capitalize on decision theory to figure out in these two environments, how does the value of the token that you currently have change as a function of time? And that's shown here on the right. For the green tokens in the uniform condition, the value of the token you currently have is always rising as a function of time because the time at which that token pays off is always getting closer and closer to you. In contrast, the purple tokens in the heavy-tailed condition have some value for the first 20 seconds, but that's largely flat, and then it drops off after that time point, because at that time point, that token basically is worthless to you. You should get rid of it and move on to the next one. So the way that we tested this was to look for any parts of the brain where there was a difference as a function of time in that region. And so we could have seen any area of the brain express any kind of difference as a function of time across the two conditions. However, we only saw one area where there was a significant interaction. That area was in the ventromedial prefrontal cortex. And the form of that interaction was exactly the form that we would have predicted. Over the first 10 seconds, we see that brain activity in this area is basically recovering from the previous trial. But then after that, you see that activity increases in the uniform condition and remains stable in the heavy-tailed condition. Exactly as we would predict if this area reflects your evaluation of the current token and how that evolves over time differentially in the two conditions. So in wrapping up, I hope that I haven't given you too negative of a view, that quitting is rational and let's all go out and quit everything now, give up. I think the point of this, rather, is to say, if we think about these kind of dilemmas the way that a systems neuroscientist would, and look for the function of the behavior that we're investigating, it actually suggests some novel ways now, once we've understand the function, in which we can reverse engineer the situation and perhaps encourage greater persistence by making situations more like that uniform condition in those places where we would like people to persist longer in the pursuit of their long-term goals. Thanks.
Hey, thanks so much. I was just curious about the token game.
Mm-hmm.
So if people know that they have, say, 10 minutes to make as much money as possible versus don't necessarily know how much time they have to make as much money as possible, does that affect their behavior in this game?
Yeah, it would affect their behavior, because the reason why you so basically we made the limited time period so that we could enforce a uniform cost of time, and if you don't enforce that, then basically the subject shouldn't quit in either condition. They should try and make as much money off the current token as possible, because there's no cost of waiting. Right, so by making it a limited period of time, you impose a cost of waiting, and we think that that's realistic. In the real world, there's always some opportunity cost for your time.
I think this was very interesting to me. Thank you. It kind of makes me think a lot about behavioral finance, and it's a much lengthier time horizon, I guess, for retirement savings. But a lot of young people are hesitant to save for retirement because it's such a distant goal, and I don't know what to really call that. Not heavy-tailed, the opposite of heavy-tailed, I guess, because you're expecting the reward to come eventually, but it's so far away you feel like you can put it off. And I don't know if you've ever looked into anything like that. Like the reward comes, it's so far away, so distant, it doesn't seem interesting now. Mm-hmm. And in reality, with compounding interest, it makes a lot more sense to do it now. Mm-hmm, mm-hmm. So why do people?
That's the exact topic that the other half of my lab studies, yeah, actually, which is how does the time at which the reward pays off affect its value? And what we know is that people discount delayed rewards. They're considered less valuable than the same reward that you get more immediately. And we've looked a lot at individual differences in discounting and how those relate to differences in brain function. And interestingly, the same area, this medial prefrontal area, seems to reflect individual differences in discounting behavior as well. And behaviorally, if you look at, so we give people financial decisions, and certainly what you see is that we're not, we haven't evolved to interact with financial institutions, right? And so, we exhibit discount rates that might make sense if you're thinking about, discounting food rewards in the wild, but don't make sense if you're discounting money that's going to sit in a bank and earn compound interest. So I think that's part of the explanation for that.
Thank you. One more?
I have a question about the part of the trials for the heavy tail distribution. Did you ever look at trials where the subject kind of made a mistake or kept waiting instead of when they should have really given up? And did the ventromedial prefrontal cortex activity look like the uniform distribution trials?
Yeah, so that's an interesting question. We don't really have enough data to look at that because we trained the subjects extensively before they got to the scanner, so there's not a lot of variability in their behavior. There's some variability across subjects, but very little variability within subjects. So we'd have to run the experiment a little bit differently at an earlier point in time to get at that, unfortunately.
Thanks.
Next, I'd like to introduce Dr. Chris Pierce. He's a professor of neuroscience in the Department of Psychiatry. He received his bachelor's degree in psychology from the University of Kentucky and his PhD in neuroscience from Indiana University, and where he did his work on dopamine signaling in the brain. Chris did his postdoctoral work at the University of Washington, where he focused on animal models of psychostimulant addiction. Currently, his lab studies the cellular and molecular mechanisms underlying cocaine relapse in animal models. And he has a highly impressive publication record, and recent work from his lab, some of you may have seen, has been featured on the Colbert Report. So let's give a warm welcome to Dr. Pierce.
So thanks, Leonardo. Thanks to all you for showing up. This is really terrific. So I really am going to talk about cocaine tonight, but the first bit of the talk is actually going to be history. It's not going to be science history, but it's going to be history, but trust me, it's not going to be what you're expecting, but it is going to be relevant. And my story goes way back to 1944, and in September of that year, World War II in Europe has reached its apex. Allied forces have liberated France, Belgium, and southern Holland. And a massive Allied assault is planned to liberate the north of Holland. This operation is termed Operation Market Garden, and at that time it was the largest airborne attack ever staged. It took place between September 17th and 25th in 1944. It was a spectacular failure. In response, Hitler punished the Dutch, who collaborated with the Allied invaders. Germany instituted a complete blockade, including all food and fuel shipments. The Dutch referred to the winter of 1944-45 as the Hunger Winter. Unfortunately, winter came early that year and was particularly bitter. Food stocks were rapidly depleted. In October of 1944, 4.5 million people in and around Amsterdam began to starve. The famine lasted until Holland was liberated at the end of World War II in May of 1945. The people in North Holland ate tulip bulbs and stripped their homes for firewood. Tens of thousands died from malnutrition and exposure. So at that time, when all the food ran out, I mean, they managed to sneak in rations from Sweden occasionally, though another problem was that the harbor froze, so they couldn't get things in with boats. But the little food that they had, the Dutch systematically rationed, and the ration was 450 calories of food per person per day. And there are drinks at Starbucks who have more than 450 calories in them. This is about a fifth of what the average American eats per day. This person is a 16-year-old young girl who survived the Hunger Winter. Her name was Audrey Hepburn. Audrey Hepburn suffered from various physical abnormalities, sicknesses throughout her lifetime, which she attributed to living through the hunger winter. And her experience in the hunger winter undoubtedly influenced what she did when she retired from being an actress. She became an advocate for famine victims throughout the world and was as an ambassador for UNICEF. So despite the suffering in the hunger winter, detailed medical records continued to be compiled. And this provided a unique opportunity to study the effects of famine on human health. And the babies born during and just after the Dutch famine have been studied for their entire lives. And this is 2 studies here. So one of the early ones in 1972 in Science magazine, and another one here from more recently in 2006. But there have been dozens of studies examining these children who were in utero at the time of the hunger winter, as well as their children and their children's children. So there's a huge database now. So a few of the observations just quickly, because this isn't directly relevant to my talk today, but the children whose mothers were malnourished only early in pregnancy had higher obesity rates, as well as other health problems, including diabetes, which is undoubtedly related to the obesity, as well as some psychiatric problems such as schizophrenia and depression. So if your mother was malnourished and you were in her uterus at that time, when you grew up, you were more likely to have problems with obesity, schizophrenia, and depression. And this isn't too horribly surprising because your mother was exposed to the famine, but so were you. I mean, you were inside her body at that time. But then more unexpectedly is these health problems, these very same health problems also plagued the grandchildren of the mothers who were malnourished in early pregnancy. So this is unexpected and might tell us about some interesting underlying biological processes, which is what we're going to be talking about today. So we think about inheritance as being a genetic process. And a genetic process obviously involves a change in DNA, which usually takes place across millennia in the Darwinian scheme of things, as I show on this next bullet point. But the environmental changes, even as severe as living through a famine might be, that's not enough to produce changes in the DNA in a person's egg or sperm, thus to make these changes heritable. In fact, there's very few things that are capable of doing that. There are some things, you know, these poor people in Japan when the nuclear power plant melted down. I mean, that's one of the examples, and there are some nasty chemicals that we know of that can cause changes in the DNA sequence, but that's about it. So most of the other things in the environment don't cause changes in the DNA sequence. So any of these changes that we're talking about in the grandchildren must be through a different process, and that process is known as epigenetic inheritance. So for the purposes of this talk, this is both the process and the mechanism. We're really not going to talk about the mechanism today. But epigenetics, just what it literally means is above genetics or in addition to genetics. Okay? So this is all a prequel to what I'd like to talk about today. So what my lab studies, as Leonardo said, and Leonardo is a member of my laboratory, is we study animal models of cocaine addiction. So this literature indicating that we might have these transgenerational effects of environmental exposures got us to thinking about, well, what about cocaine? What about drugs of abuse? I mean, might they influence the offspring and then the grand offspring and generations beyond that? So we studied paternal cocaine exposure. So the dads took cocaine, and we looked at learning and memory in the offspring. So here we have the setup over here on the left. So this is the standard male Sprague Dolly rat in what we call an operant chamber. And up here we have a syringe that's full of cocaine. I don't know if you can see it, but there's a line that goes up like this, and it goes down to a port between the guy's shoulder blades. And from that port, the catheter goes over his shoulder into his jugular vein. So we have a direct line from this syringe of cocaine to the animal's heart. And we allow this animal to press this lever to receive cocaine. Now, how hard do you think it is to get them to press that lever for cocaine? It ain't hard at all. We put them in the box, we close the door, we don't do anything, no kind of pre-training. They explore around the box, they bump into the lever. Once they get a few shots of cocaine, they know it, they like it, they press a lot for it. So in this experiment, we have the cocaine dads and we have the saline dads. So this is the cocaine self-administration behavior, and these guys self-administered cocaine 2 hours a day for 60 days. How much cocaine is this? It's a lot of cocaine. So as a matter of fact, we have to regulate their cocaine intake because the advertisements you see on TV are true. If we left these guys to their own devices, every single one of them would overdose on the cocaine and die. Every single one of them. The saline control animals, every time this guy received an infusion of cocaine, there was another box down here, and they had a guy set up just like this, but hitting the lever did nothing, and just every so often, when this guy received cocaine, this guy down here received saline. So that was our control group. Okay, so we took these cocaine guys, and we took the saline guys, and we mated them to naive females, okay? So in science, so these are the parents. So we call them the parents, scientists call it F0, the F0 generation. So the cocaine guys and the saline guys were with the females just long enough to impregnate them, and then the cocaine guys went away and were never seen again, just like cocaine addicts in real life. So the females got pregnant, they had their babies, and the babies grew up, and then we asked the babies various questions. Well, first off, what do we call the babies? So this generation, the parents are called F0, and scientists call the children F1, the first generation. The children is F1, and if your dad took cocaine, we called you cocaine-sired. If your dad received saline passively, we called you saline-sired. Once these guys grew up and reached adulthood, we asked them, well, we didn't ask them, we looked at learning and memory problems in these guys. And we used two different tasks, one of which is called object place recognition, which is dependent on an area in your brain known as the hippocampus, and another one called novel object recognition, which you don't need a hippocampus to do this second task. This down here, this is two stylized images of the hippocampus. It's even when Golgi looked and Cajal looked at it, it was a beautiful structure just with Golgi staining. This is a artistic view of it, which was done by a former neuroscience student here at Penn named Greg Dunn, D-U-N-N. So if you would like to buy one of these images, this is what he does for a living. So gregdunn.com or something like that. But I warn you, they're not cheap. So, but they're very beautiful. So the hippocampus is, not surprisingly, a brain region that's critically involved in learning and memory, particularly spatial memory. So if you are pathologically losing your keys, you might have a hippocampus problem, okay? And then more seriously, people like Alzheimer's patients, pathology in the hippocampus is one of the first places it shows up. Okay, so we have two tasks, object place recognition, which depends on the hippocampus, and novel object, which doesn't. Okay, so what's this task? So the first thing we do, we just have an open field. We put the rat in there, allow him to run around for a little bit, habituate to the chamber. The next day, we put him back in the same spot, but now we have two objects which are absolutely identical. And we allow the animal to explore these two objects. They spend about the equal amount of time exploring each of the objects. Then the next day, we move one of them. We take this one, we move it over there, and the animals tend to explore the one that's moved more. Okay, so we've moved something, the spatial orientation of these things has changed. We said that the hippocampus was involved in spatial memory, so not surprisingly, this is hippocampal dependent. Novel object is similar, except for training, we've got the same two objects, but then during testing, rather than moving one, we just swap it out for a novel object, okay? And they tend to prefer the one that's novel, right? And you don't need a hippocampus for this. Okay, so then we ran our offspring of the cocaine dads and the saline dads through these tasks. And first, let's talk about novel object recognition. So if we look at the white bars, this is training. So this is during the period where they just get to explore these two objects. And I told you that they spend about the same amount of time exploring the two objects, and that's what we see, right at 50% through all the groups. Then the next day, we swapped out one of the objects, and I told you that they tend to prefer the novel object. So in this case, in every group, they preferred the novel object, okay? So the blue is for boys, and the pink is for girls. So this is, in every case, this is normal behavior, okay? So none of these animals, their novel object recognition is just fine. That's in contrast to spatial object. So again, females, so unlike Teresa's talk, in our case, the females are just fine. They do super. But the males is a different story. So in terms of the spatial object recognition, it is completely gone. So they have no preference of one for the other. They have no recognition that that object has been moved. They spend equal time. So we have a severe deficit in spatial object recognition. Okay. So the neuroscientists in the audience, if you saw this data, well, the first thing you'd say, well, everybody in the audience, hopefully you're thinking hippocampal deficit, right? Because I told you that this task was a hippocampally mediated task. all the neuroscientists in the audience then undoubtedly immediately thought, let's look at LTP, which is what we did. But first, I should tell everybody else, what is the LTP? So LTP is something called long-term potentiation. Long-term potentiation happens to have been demonstrated the year I was born, 1966, 47 years ago. So LTP is an enhancement in neuronal activity produced by electrical stimulation. So we put a little electrode down in the brain, we stimulate these electrodes, and the electrodes cause a change in the activity that's persistent. So changes in the brain, that's what we get with learning and memory. Learning and memory, by definition, results from a change in the brain. And LTP is widely considered to be one of the mechanisms underlying learning and memory, and not surprisingly, one of the easiest places to get it in the brain. And the first place it was demonstrated was in the hippocampus. Okay. So here we have in white, we have the saline-sired guys. In blue, we have the cocaine-sired guys. So over here is baseline. So during baseline, the cells are just firing along at their normal rate. And then here at theta, this is something called theta burst stimulation, but this is where the electrical stimulation came. So in the control group, then we get the potentiation. So this is the LTP response. Okay, so we have an enhanced neuronal response and it stays up there. So we can think about normally during learning and memory, you're trying to learn something, and then during this, this process, you're consolidating those memories, and the hippocampus is deciding what it's going to remember, what it's not, and then it's going to store those memories. So the critical thing here is it is completely absent. If your dad took cocaine, you are incapable of spatial object recognition or hippocampal LTP. So this tells us, first off, it confirms that yes, the hippocampus is screwed up, and this is a physiological correlate, we call it, of the behavioral response that we observed. Okay, but I told you that changes in this case, the changes in the offspring are not that surprising. So in this case, you can imagine the dad took cocaine, but his sperm is also seeing the cocaine. And those sperm became these animals that we're testing here. Not so in the next generation. If you look at the grandchildren, they have never seen cocaine at all, in any way. The sperm that created them did not produce cocaine. So the next question is, what's happening with the grandchildren? So these are the parents. These are the children we just talked about. The girls are just fine. In both cases, if your dad took saline, you're just fine. And this guy here is the one that has the deficit. Okay, so now we're going to look at the grandchildren. And the critical groups, we just showed you these two data, and I'm going to show them again, data from these two groups, the cocaine-sired and the saline-sired male. And then in the next generation, the grandchildren, we're going to, if your dad took cocaine or your dad received saline. And basically, we show that the phenotype, and phenotype just means that the characteristic that we're observing is both in the children, so severe spatial object recognition deficits that are also there in the grandchildren, okay? So then, naturally, the next question is, what's happening with LTP? And this is a bad way to wrap up a talk, but we don't know yet. So our prediction is that they would have LTP deficits, but these are unpublished data. It's hot off the presses, so we don't know the answer to that question. But the bottom line here is that if you take cocaine, if you're a male and you take cocaine, you can be influencing the behavior and physiology of your children, and not only that, you're going to influence the behavior and physiology of your grandchildren. And who knows, it could go beyond that. My prediction that it will. But we don't know the answer to that last question. So as I said, Leo's in the lab. He was involved in this project. The lead author on this project will be Matt Wimmer. There he is. And everybody in the lab's got kids these days. Thank goodness I'm not having any more. These experiments scare the crap out of me. So everybody in the lab contributed, and I thank them, and I thank you for your attention.
Do you think that, like, as you said, that you think it's going to affect further generations, but do you think that the effects on the LTP will be as pronounced continuing the generations? Do you think it's going to kind of like maybe normalize or meet someone in the middle?
Well, so so my prediction with the the grandchildren, I expect the LTP to be gone because the behavior's gone. So my expectation is that we will see the LTP gone in the grandchildren. And then the grandchildren, that indicates that there's a change in the germline, to get more specific about it. And I mean, if there's a germline dependent change, my expectation is it's going to be there with the great-grandchildren too. But you never know. I mean, these things could fade away, but the grandchildren data indicate that we do have a change in the germline, so it's heritable. So if it's heritable, I think it's still going to be there the next time.
Do you envision any future studies, or have read any that are maybe doing this already, regarding the actual dose-dependent effect? Like, how much cocaine does dad need to do to actually cause the effect, and what's the lower threshold to see these effects?
Yeah, it's a really good question. So in our lab, we study addiction, and most of the time in the lab, we study, as Leo said, animal models of craving and things. So we really think that the self-administration model is very close to what humans do, because as I said, we don't have to convince these guys to take cocaine. So that's a long-winded answer of saying I think we did really overkill in this case. I don't think that the animals had to self-administer the cocaine. I think if we put the cocaine in a syringe, administered it to the animals, we would have seen the same thing. Because the critical thing in the dad generation is just that the sperm saw the cocaine. I don't think the brain's involved at all. I don't know that for a fact, but that's my guess. And so I think that we made it too complicated, but the amount of time is a good question. And I don't know. I mean, so 60 days is the length of spermatogenesis, which is why we chose that. We were very conservative. If sometime in the future we find out that 7 experimenter-delivered administrations of cocaine are enough, all my students are not going to be happy with me because it makes it a lot more difficult.
How specific do you think your effects are for cocaine? Would this generalize to other drugs or other addictive substances such as sugar or chocolate or coffee?
Yeah, I'm not touching those last ones. But there's clear evidence that this is new stuff. What we've been doing primarily is looking at the reinforcing effects of these drugs. So we do, the dads take cocaine or nicotine, or other labs have looked at things like heroin, and then they've looked in the next generations and seen the influences on drug-taking behavior. And it's too complicated to get into, but there are definite effects. They're not all the same. So not all these drugs of abuse don't all produce the same effects, but they're all producing transgenerational effects, including nicotine. And I've got bad news: if we have drinks afterward, it's alcohol too.
We'll have to take other questions for afterwards. Thanks again to that. Okay. Finally, I'd like to introduce Dr. Douglas Smith, who is the Robert A. Groff Professor of Neurosurgery here at the School of Medicine and the Director of the Penn Center for Brain Injury and Repair. Dr. Smith has devoted his full-time efforts to the study of neurotrauma following completion of fellowships both in molecular biology and neural trauma at the University of Connecticut. His research interests have included mechanisms of nerve trauma and repair, magnetic resonance techniques for the diagnosis of diffuse axonal injury, traumatic brain injury, and cognitive dysfunction, and the link between brain trauma and neurodegenerative diseases like Alzheimer's. Dr. Smith's research efforts have resulted in over 160 publications, and his work and expertise have been featured in a variety of popular media sources such as the Scientific American magazine and the documentary Head Game. Give a warm welcome to Dr. Doug Smith.
Well, it's really great to be here. And I want to talk about a device that's really unique that we want to emulate if you think about brain-machine interfacing. I don't know if everybody can see that, but this is really just a toy. The device I want to address is this thing, because that really should be the goal. And I'm worried that, sometimes some of these efforts that you see that are spectacular to help people who might be locked in may fall short of getting to the hand, or the human hand. So, it's not advancing. Is there a clicker or something? Yeah, it's not going down. Okay, so I have disclaimer or disclosure that some of the technology I'll talk about has been licensed by Penn to a startup company. And to begin with, have you ever wondered, I mean, when the iconic scene from Star Wars occurs, there's a pinprick on this hand, it moves after Luke has his arm cut off, and that means that a signal from this prosthetic has gone to his brain and a signal back that his hand twitches. And I'm really curious about what this thing is, because what's going on there? I mean, can you poke something hard into the nervous system and it'll like it? Your nervous system did not evolve to get hard, sharp things jammed into it. It's evolved to accept something wet, kind of familiar, and it should be nervous tissue. And I think we're at a crossroads right now that's akin to when over 100 years ago, people were trying to decide, or really there was a huge debate, about how to bring electricity to our homes. And now we have to decide how to bring electric currents from our body to drive devices and how devices can communicate with our nervous system. But the state of the art is not really great. This is actually the most widely used multi-electrode array. It's put into the motor cortex, and then you have neurons there that have to learn how to kind of spike to get these different regions to record from them. Now, the problem is that your motor cortex doesn't actually run anything. It just tells other people what to do. So this is limited. It's great, like if someone has very severe ALS or is paralyzed or quadriplegic, but the movements are somewhat gross. And you might be able to move a cursor on a screen even, or move a limb that looks real, but it cannot pick up a dime. To pick up a dime, there is a lot going on. For every nerve, large nerve in your arm, you have about 1 million nerve fibers or axons that will direct that movement. And it's not just moving the motor component, the muscles, it's actually getting the sensory feedback. And those two together combine to form proprioception. Without proprioception, you cannot button your shirt. You cannot catch a ball. You have to look at a hand and just say, okay, hand, move this way or that way, and it could be very disabling. You will never regain that true function that a human hand can accomplish. So we've created something that kind of looks like this ugly creature. I think it's beautiful, but it's not a jellyfish at all. It's an engineered nervous tissue, a nervous tissue construct that is almost like developing a mini nervous system in culture. So a mini nervous system is really what your nervous system wants. It likes the wet stuff. It likes the neurons. It will embrace them like old friends, old relatives. Right now, as I'm speaking to you, little nerve fibers, dendrites, and axons are finding new targets, and they're welcome no matter where they go. If I transplant a neuron in your brain, very likelihood it will survive and be embraced as a new friend and neighbor. But as far as the nervous system taking on electrodes, even flexible ones, this is foreign. It wants to fibrose over, scar over. Maybe it wants to reject it. Maybe it wants to just die because it got pierced. So what we're looking at to develop this technology is something for you neuroscientists in the room, which I assume most people are, and most people have the understanding that you've learned in the first chapter that the cell body extends an axon, that large nerve fiber. There's a growth cone that limps along and finds a target. If it's an embryo, that's about 2 millimeters away. And then there should be a synapse, and the next chapter is on receptors. But there's something that's been missing from the textbooks, and that is what happens after synaptogenesis or after integration of that axon. Something huge can happen. In fact, something maybe far more dramatic, maybe far more impressive than that first phase of growth. That's that second phase. It's implicit. We see it everywhere around us. I mean, how does a giraffe have axons, or a blue whale have tens of meters, 30 meters of axon? He had a neuron in the brainstem that connected to a motor neuron in the tail. Again, about 2 millimeters away, but the animal grew 30 meters. Those were mechanical forces that caused that growth. So those mechanical forces, like the growth of vertebrae, will add material, and the axon, there's a mismatch between the tissue properties. The tissue stiffness of the axons in a spinal cord are not that great. So they can either go along for the ride or get stretched down like a rubber band until they snap, but they don't. The blue whale does great. So this kind of ignored area of neuroscience is a really important area. It has been ignored, but can be exploited, I think, for repair of the nervous system. So the idea is just to copy that. We built a lot of really weird gizmos where we have a box. We can grow two populations of neurons next to each other on a top membrane and a bottom membrane, and use a microstepper motor system to separate them after that first phase of growth has occurred, and axons span those two populations. So basically, just as a top membrane, bottom membrane, there's integration between, and we just turn on a box that stretches further and further. And these axons that span those two populations don't thin down. In fact, they grow thicker in diameter than if we did not stretch them. This is a natural process. And they organize themselves in very large bundles, otherwise called fascicles. So the growth is impressive. It goes on and on and on. We've gone a centimeter a day. We could probably go more. We've gone 10 centimeters in length. We could go probably across the room. The blue whale obviously can do quite a bit. And what we see is that these axons will organize themselves into larger and larger bundles, coalescing into these large fascicles. And over time, you end up with something that is more and more organized, which I think recapitulates ontogeny, recapitulates what happens during development. You might have somewhat random trajectories of axons spanning two populations of neurons, but over time it becomes highly organized into tracts or nerves. In our case, we see something that looks almost like harp strings or beanstalks that can occur over time. And this can be done with almost any type of neuron. Motor neurons, sensory neurons, human, animal, doesn't matter. Every neuron seems to have this capacity for this enormous, we call it extreme stretch growth, that can grow beyond the dogma. I don't know if anybody studies neurofilament protein transport here, but that should be limited to millimeters a day. Fast transport can barely keep up to the pace that we're looking at here. The blue whale exceeds everything. So all this type of growth goes against the dogma that we've all been taught in textbooks. But despite that growth, things can maintain function. We can stimulate one end, record from the other of this construct. So we've been very successful, I think. We can grow things really fast, really long, maintain their geometry and function. And yes, we have the world record. We're kind of the only ones in the race, so that doesn't count. And we may not actually be the first people to think of something along these lines, as we heard different. It's just so strange that people want to do all kinds of things like this, but not necessarily with the same goals. For us, we want to transplant this material. Now, it's nice to be able to do this in culture, but it has to come out of culture. We've created a three-dimensional geometry, very organized geometry. And I don't know how well you can see this. See these kind of lines right here? Those are axons. There are about 1 million axons there in kind of this planar structure. Well, we can embed a hydrogel like collagen or whatever and lay it down. It links up, and then we can peel it out, kind of like using the silly putty on the comics, And we can take this out of the culture environment, and if you've ever had a fruit roll-up, we can then roll that up and put it into a tube, thereby creating an entire construct. Now, we've used this to repair spinal cord injury, which I'm not going to discuss today. We've actually used it to repair the brain, various brain lesions. But I will briefly discuss peripheral nerve repair, where picture a deficit, a lack of a nerve between a large gap in a nerve. And when you cut the axons on the proximal side, they're completely gone downstream. So you have two stumps, but the distal stump has no axons in it, and therefore you get no innervation to your limb. But when we transplant this, we can go up multiple centimeters of a transplant. If we put just a tube between the two nerve stumps, axons from the host would never go centimeters across that tube. That will never happen. But when we put the construct in and wait several months, we end up with something that looks on the outside exactly like the nerve that used to be there, but that was a gap at least 5 centimeters long, and now there's a complete nerve on the outside. When we look in the inside, we still see the cells we transplanted here, these large round bodies. You don't have neurons in your peripheral nervous system. This is what we transplanted there. The green are the transplant axons, but down here on a larger scale, we see red coming in. Those are the host axons coming in, and the host axons love the graft axons. They love them so much that they intertwine with them. They know what they are. This is what they do. They will follow those other axons to the complete, very distant end of that transplant region because they have something to follow. It's axon-induced axon growth and guidance. So the axons themselves provide perhaps the strongest signals to entice the host axons out across this huge gap to then reinnervate and restore function in that limb. And we've had really wonderful results in a large animal model, and we're hoping to go to clinical trials in a few years. And even over time, we see myelination. If you look at cross sections of these, the outside red is myelin, inside green is the axon. So this has been very exciting, and we're very pleased that we have something that might have a clinical translation soon. But for the purpose of a brain-machine interface, we think that this is also one strategy that we should pursue. So think of it, rather than just bridging damage of a nerve, if the whole arm is cut off, the nerve stump is still at the stump of the arm. It still has all the axons in that stump. So if we can cobble together the same way we just did, at least on that end, with repairing the nerve, but on the other end have a multi-electrode array that might be able to interface with the device. So think of it where we can actually, while your nervous system doesn't like to be poked and jammed with some kind of electrodes, it doesn't know what to do with it. If we grow neurons selectively, decorate these delicious chemicals on the electrode array, the neurons will adhere very tightly and will survive there and stay there, maintaining their geometry post-transplant. So in one iteration, we use this array where we have, this is the outside that can be attached to the device. Over here are the tips where we have all the electrodes shown here. We can see the cells on these electrodes and then put them in what we call the axon elongation device. These are the shanks, you can see they're the darks and that go on. These are the axons, these long striations that go all the way across to the other populations of neurons. So this would be the end that we would transplant to a nerve stump, and that these axons would extend out to these shanks. And notice that there's a preferential growth of the axons at the tips of the shanks. I mean, we didn't even expect that. That was a definite bonus. Again, the shanks are where the neurons are, and you can see all these green axons going out that will travel whatever distance we need to separate the device from the nervous system. We can use fancy techniques to use light channel rhodopsin, in this case, to use light to stimulate one end and use the multi-electrode array to record from the other. And we have 1 million axons, so we can have, this is a smaller array. Each box is an electrode, but we can stimulate some with light and just see the response at different regions spatially. But this could go up geometrically, the number of responses you need. To pick up that dime with your fingers, you are getting that two-way communication. You're feeling the edge, you know where it is, you pick it up, somebody throws a ball, you catch it. We need that kind of, that incredible depth of information. And what this also allows us to do is that two-way communication. So it's kind of like can ping-pong back and forth. Stimulation can be made at the multi-electrode array, travel down the axons to the body. Body can send a signal that could drive a motoric kind of signal to drive a device. And again, that two-way communication is the only way we're ever going to be able to button a shirt or do simple tasks. So again, the same iteration as you see in the peripheral nerve repair. The difference is at one end, there's not a nerve. It's just where the arm stump might be, where we have the multi-electrode array and the connector that can go to a device. We have the neurons and the long axon tracts in the middle that are then cobbled to the host nerve stump. The nerve stump then will send out regenerating host axons that will intertwine and follow the graft axons, because that's what they do. This is kind of a backdoor approach, kind of a trick to, rather than worry about shoving an electrode into the host, you're enticing it to come right out and join the electrode directly. And that's what we see. So when we transplant, again, the green is the graft, and the red are these axons coming out right to the electrodes. They've been drawn, kind of the siren song from the graft axons are bringing them right to where we want them to be. So what we can see is that you think of an iteration where whether you want a hand transplant or full arm limb for a prosthetic, these red lines are nerves kind of blown up over here, that over here there's the elongation of the axons and the neuron bodies at each end. So you can picture that this wet human nerve is sending out the wet axons that will grow across the wet interface. That's what they want to interact with. And then in the laboratory, we have, in this case, depth electrodes, where those axons can then be brought out to various regions of these electrodes for any number of connections to drive very, very, very complex devices. So this is what I'm talking about for the Luke Skywalker. So I would like to think that spot right there was really where his fibers, these red fibers, could go across, reach across the shanks till they finally are brought right up to the electrodes. Now, I've been a little remiss. I didn't put in the slide with all the huge team of people who have worked on this, so I'll just take the credit. Thank you. Thank you very much.
Very fascinating talk. I was curious about the peripheral nerve system, when it regenerates, can have a lot of improper matching with motor and sensory nerves. I was wondering if you looked at some of the functional connectivity in animal models with your graphs.
Yeah, so think about it this way. So people have lost hands completely, and you put the hand back on, their own hand. Sometimes people had transplant from an organ donor even. There is no way those axons are going to back out to the same muscle units. And yet they go in a very diverse direction, but through remapping, it's amazing that the motor system can remap so quickly in your brain that they can quickly learn to regain function of the hand. Sensory does not remap quite as well. And the really issue for any kind of transplant like this is a thought about pain, where a sensory fiber might join to, let's say, a muscle unit. But surprisingly, it seems to make sense. It's definitely not going back to the original place, but it's going to a place that, by and large, does make sense.
Well, I had a very similar question. But it was about people that had a spinal cord injury. So if you can, just put this thing in between the two severe ends, and then, perhaps you can restore some of the function. And the other one was, with this thing of the elongation of the axons, you said that they get thicker, and presumably they also, the time it takes for a signal to travel through them gets shorter. I mean, if you get a thicker axon, it conducts faster. So are you measuring that? I can even think that perhaps that's sort of conserved or compensated for.
Yeah, so we have. So some of the biophysics, let's say sodium channel biophysics, they do have higher sodium channels, much like children have more sodium channels on their axons because of constant remodeling of the brain and whatnot. So they do, but their conduction velocity is the same as you might predict for whether it's myelinated or unmyelinated, but the thickness can have an effect. For the spinal cord repair, there are two ways to look at it. The first way is the most likely. If we use nerves of peripheral origin and create these constructs, they look like the kind of, you might know of the Aguayo experiments, they put a peripheral nerve in. But in this case, we can entice motor fibers to come across what really is a vast cyst, which often forms in spinal cord injury, and follow these constructs all the way down to the other side. And in one case, we've seen that they have formed a new intraspinal circuitry. So the other more elegant, or more, I guess, more exotic type of thing is that these constructs can act like a relay themselves, so that the host might synapse on one end, and now the signal can be propagated through the construct itself. So,
hi, thanks for your talk. This is kind of a wild question, but you could envision having supernumerary limbs, right? You could envision having multiple different, like 5, 7, 8 hands. So just thinking at that level, if you want to increase function, is what's the limit on the system that you're working with, and the remapping that can occur in the cortex to
That's a great question. I think that especially the young people here, your grandkids are going to ask you, say, did you have to use a keyboard? So I think forget limbs. I think there's going to be just everything. Can you imagine just changing channels or whatever? I mean, this is going to be driving vehicles. Right now, the DoD is very interested in this, rather than take all the time with that toggle switch that you just think this. So it's not just simply limbs. I mean, the sky is the limit. There is nothing that what we do with our hands to manipulate the environment that couldn't be done even just perhaps in our minds.
Just before we all head out, I'd like to take a brief moment to say a few words of thanks. First of all, I'd like to thank all the speakers here for their time and their effort to be here with us, so you can give them a round of applause for them. I'd like to thank the Mahoney Institute of Neurological Sciences and the Biomedical Graduate Student Association for their generosity in supporting this event tonight. I'd like to thank John Danny, Josh Gold, Jane Hoshi, Jackie Foltz, and Kelly Jones-Shudo for their invaluable administrative assistance with this. I'd like to thank the Public Lecture Planning Committee for organizing the event, with a special shout out to Dave Reiner. Dave, without you, I don't think we could have made this night happen. So. And last but not least, I'd like to thank you all for braving the cold weather to come out and be here with us tonight. We have a reception just outside. We encourage you to stay for the food and hopefully sparkling conversation that follows. So thank you very much for coming. Have a great night.